So here we are close to a second restored Rhône, second restored oxbow. So this is the so-called Rhône de Sislande. So as we said earlier, it is now disconnected from the main channel. But since we are higher than the reserved discharge, there is currently water in this oxbow. As you can see just behind, there's also answers Stanford's question earlier on. So the Pierre-Benite site, as Johan said, it was one of the first restored sites for the Rhône River. It was also one of the first sites where research teams implemented scientific monitoring. First of all, there have been modelization works, and the increase of the reserved discharge was discussed together with scientific teams based on habitats models that I mentioned this morning during the presentation. The goal was to identify a discharge in order to have a positive answer, positive reaction from fishes and macroinvertebrate communities. The second objective was to collect data in order to validate these models. So the first monitoring projects, so that there was a whole series. So this is a chart as I've shown this morning, but it's only for this site of Pierre-Benite, although this morning was addressing the entire Rhône River. So the first monitoring that were developed on this site were the monitoring of fish species that were implemented in 1895. So in 26 years, there were 4 measurement campaigns before the restoration works. And the other monitorings that were developed addressed macroinvertebrates. So 4 years of monitoring, 1 year of restoration. There you can see the before and after. And then the monitoring was performed on a regular basis for over 25 years. Regarding macroinvertebrates, there you can see a picture showing the sampling points, the sampling steps. So this was made on sampling points in the channel. So you can see the blue dots that are the various sampling points, and that was reproduced at first it was yearly, and then we gave longer time between each measurement campaign. And usually it takes place at spring and in the summer. And here you can see the results of these monitorings. So the same as the diagrams, the charts I've shown this morning. So with the situation of invertebrate populations who enjoy before the works and after the works. So there you can see the benefit of the increase of the reserved discharge quite clearly. It's one of the sites where it worked pretty well because the discharge was significantly increased and multiplied by 10 on this site. Regarding fishes, that's the same principle. So there you've got electric fishing that is performed in the Rhône channel. So from a methodological point of view, it's extremely complicated to do sampling in the Rhône River. So this is electric fishing from a boat or walking directly in the channel. And in the same way, you've got various sampling points distributed all along the line. Just to give you an idea, statistical works, or so there have been statistical works to show the accuracy of these monitoring works, and it has shown that if the fish population is multiplied by 2, with that method, scientific have a likelihood of 5% to notice that kind of change. So it means you need a huge, meaningful change to have a chance to notice this from a scientific perspective. So changes were quite important here. As you can see here, the changes before works and after the works. Same on fishes that appreciate current or running water. So the trend is quite significant on this site here compared to other sites. Another monitoring that was performed is that of sediments in the backwaters, so in the oxbows. So on the picture, it's the Rhône where we are located here. So the principle here is to have measurement points of sediments throughout the line of this oxbow. So we are trying to measure the thickness of fine sediments that were deposited at the bottom of the oxbow yearly. So this monitoring is performed every year in order to assess where the sediments dynamic in the oxbow, and it enables to produce models regarding the life cycle of the oxbow. There again, it's very time-consuming to perform such monitorings, and especially environments that are not easy to access. And as Johan said, on Cislon here, so this is monitoring that is performed yearly, and the life cycle, so the life duration of the Cislon oxbow was quite low in terms of sediments, but there was an external factor with the insertion of the channel and the moving of an apron inside the channel, and basically this disconnected the oxbow. So the restoration that was set for a specific hydraulic operation of the old Rhône River is not operational anymore. And that's why it's very important, as we said this morning, it's very important to take into account the entire context when designing that type of works. And there you can see the various topics that are monitored on this site. So these are scientific monitorings. And as I said this morning, we have more and more narrow collaborations between research teams, site managers like SMIRIL, CNR, and engineering design officers in order to collect data, to pool efforts on the field, because it's very difficult to collect data, and also to pool analysis efforts and expertise in the way we discussed the obtained results. And in this site, there are even campaigns that are co-led by site managers and research teams, namely in terms of macrophytes, water vegetation, aquatic vegetation. So Samuel is in charge of supporting the monitoring from the SMIRIL site. Just to add on regarding the monitoring aspect, this morning you heard about the articulation between scientific monitoring and the monitoring that we implement as river managers in order to assess the efficiency of the restoration works. So these are large-scale restoration works, as you could hear. It's not only on that site. And this is the illustration of how the scientific monitoring really addresses the aquatic aspects, as presented by Bertrand. The site manager that is mostly addressing global indicators of ecology and biodiversity in the environment. And we also provide the engineering vision, the feedback and experience, and we try to draw a link to establish a relationship between all these elements. So basically the aquatic monitoring is the answer to the works that Johan will present just after that. So basically we operate on the Rhone environment. We remove the former works of the 19th century. We reopen the backwaters. And all the issues introduced by the SMIRIL regarding the alluvial strip associated to the river and that forest is basically pushing us to take a step back and not only focus to the purely aquatic environment, but to really address the global operation of that system. And this operational monitoring that is basically undertaken by the CNR relies both on the past and future scientific works. Whenever possible, it also relies on the site manager works, and when there is no site manager, we basically compensate for that, and we perform the monitoring works instead of a manager that is not available. And the second aspect that relates more to the global biodiversity of the site is a requirement in terms of regulatory monitoring, because under French law, when you perform this project, even if the aim is ecological restoration, as you can see, this is heavy works, and basically you intervene or you have an impact on an existing biodiversity, and even if it was degraded by historical developments, you need to take a whole corpus of species into account. And the state requires us to implement monitoring measures in order to assess the impact, both positive and negatives, of our works. So we have to monitor the development of the communities over a very long period of time. So communities like amphibians, insects, and so on and so forth. So this is closely, there's a close interrelationship between scientific monitoring, engineering works, and feedback to see whether it works or not, or make adjustments, and regulatory monitoring. And the real challenge is really the real challenge is to have enough cross-cutting expertise in order to interpret the various scales of monitoring. So basically taking a step back from a specific expertise and going beyond that. So maybe this or that species of insects or fish had a positive reaction, but another species will not have such a positive reaction. So what should we conclude? Was it worth doing that project or not? So basically, it's all about being able to take a step back and to assess the global ecology of the entire territory and to make conclusions in terms of biodiversity.